So hello guys, we will be starting our new session about photosynthesis. So basically our next scientist is T.W. Engelmann. Is T.W. Engelmann. T.W. Engelmann took a prism and split the light into different components of its wavelength, and by illuminating the algae named Cladophora, he obtained that for different wavelengths of light, different photosynthetic activity is being observed. Okay, so what he has done? He has took a prism, okay, and he split the prism light. He split the sunlight into different components in different lambda or different wavelengths of light to a green algae named Cladophora. Okay, so here illuminated a algae named green algae named Cladophora by separating the sunlight into different types of wavelength. Okay, so what he has obtained that, so basically suppose he has split the light into red color or into green color or into violet color. Okay, so basically he has seen that the plant will show different photosynthetic activity. Okay, different photosynthetic activity. So what he has done, talking about the experiment done by T.W. Engelmann, he has taken a beaker, a flask or beaker, whatever you may call it, and he has sunlight here. He splitted the sunlight into different components of wavelength by splitting it into different component of wavelength by prism. Then he illuminated a algae named Cladophora, which is shown in green in color here, and he has he has placed aerobic bacteria above the solution. Okay, so he has placed aerobic bacteria above the solution. Okay, so what is the role of aerobic bacteria here? The aerobic bacteria, if the O2 concentration increases, the aerobic bacteria will also increase. So basically this aerobic bacteria serves as an indicator of O2 here. That means O2 is directly proportional to aerobic bacteria. That means if the concentration of O2 is high, then the aerobic bacteria will grow, and the concentration of O2 is low, then the aerobic bacteria will fall or will not grow. Okay, so this is the equation here. So basically what he has observed that for red and blue algae, for red and blue wavelengths, for red and blue wavelengths, algae shows maximum photosynthetic activity. Okay, for red and blue algae, the algae shows maximum photosynthetic activity. Okay, that means for red and blue light, the photosynthetic activity is maximum. He had seen it experimentally. Okay, this is the experiment done by T.W. Engelmann. If you didn't understand the experiment, please go through the video again, we'll understand it. So what was the conclusions drawn by him? This conclusion point will be very long, because in this conclusion point I will be explaining the action spectrum and the absorption spectrum here. So in the conclusion, he has in the conclusion, what is the conclusion drawn by him? The first conclusion drawn by him the first conclusion drawn by him was in red and blue region, maximum O2 is being produced. We have already seen it. And for chlorophyll A and chlorophyll B and chlorophyll A and the chlorophyll B are the crucial pigments for the photosynthesis. How he has proved this notion that chlorophyll A and chlorophyll B are the crucial photosynthetic pigments, we'll understand it in very short time. Okay, so do not worry about that. And the most notable work of T.W. Engelmann is that he has obtained the first action spectrum among all. Do not panic. I will be explaining what exactly is the action spectrum. Okay. So T.W. Engelmann first obtained and explained the action spectrum. Okay. So we'll be first explaining the action spectrum. Okay. So what is action spectrum? So basically, action spectrum. Okay. So basically, action spectrum. Action spectrum is relation. Action spectrum is relation of relation between O2 produced and O2 produced and wavelength where the photosynthesis is occurring. So action spectrum is relation between O2 produced and wavelength in which the photosynthesis is occurring. Okay, so action spectrum is so what is the function of action spectrum? So action spectrum gives O2 production rate in a given wavelength. Okay, so action spectrum gives O2 production rate in a given wavelength. So suppose so we'll be talking about action spectrum. So it is in the x-axis we have lambda, that is the wavelength on which the photosynthesis is occurring, and the y-axis we have O2 production rate. Okay, O2 production rate. Okay, so what we have obtained, the curve is like this, this, and this is how the action spectrum looks like. Okay, please see your NCRT, the curve is given there. This is how a action spectrum looks like. If we are to determine O2 production rate for a particular wavelength, we will draw a perpendicular from the y-axis, from the x-axis. X-axis, suppose this is lambda 1, and we have to determine the photosynthesis rate, O2 production rate in the wavelength lambda 1. We will draw a perpendicular in this curve, and where this curve is intersecting, we will be perpendicular, drawing another perpendicular to the y-axis, and where this line is intersecting, suppose this is A, this A will be O2 production rate at lambda 1. This is our conclusion. Okay, that this A will be O2 production rate at lambda 1. So basically this curve which is drawn between O2 production rate and the wavelength in which the photosynthesis is occurring is known as action spectrum. Action spectrum. Okay, in the next in the upcoming graph we will be explaining the absorption spectrum. The absorption spectrum is just like the action spectrum. In the action spectrum we have taken O2 production rate in the y-axis, and in the absorption spectrum we will be taking absorption of photons for a particular pigment in different wavelengths. Okay, so see here, this curve is pretty much bad shape, so we cover. So let's take the next slide. Let's take into the next slide. Okay, so this is absorption spectrum. Will be this is x-axis, this is y-axis. In the y-axis we have the photon. We have taken the photon, the photon absorption rate, and the x-axis we have taken lambda, or the wavelength in which the photosynthesis is occurring. Okay, photon absorption rate for a particular pigment. So we have to understand that different pigments of the plants shows different types of photon absorption rate. Okay, so if in a particular wavelength, if chlorophyll A absorbs certain photon, uh, in the same wavelength, suppose lambda 1, the chlorophyll B will absorb another amount of photon. Okay, so we will be, so the absorption spectrum or the photon absorption rate, this versus lambda graph will be drawn separately for different pigments. Okay, so there will be one graph for one pigment. Okay, so we'll be drawing the first graph here. We'll be drawing the first graph here between chlorophyll A. Okay, chlorophyll A. So our first graph is, this is chlorophyll A graph. Okay, chlorophyll A graph here. This rise down. Okay, this is the graph of chlorophyll A. You have understood the graph already, that this is the photon absorption in the y-axis, and the x-axis this is wavelength, or which is represented by lambda. Okay, and this is graph of chlorophyll A, and the graph of chlorophyll B is just like this. The graph of chlorophyll B is just like this. This, this, this is the graph of chlorophyll A. Chlorophyll B. Okay, so what I was talking earlier, that suppose this is lambda dash, arbitrary wavelength they have taken, and we have to find the different photon absorption rate for this lambda dashed. So we will be drawing perpendicular to chlorophyll A, and from the intersection point we will be drawing another perpendicular to the y-axis. So this is suppose A1, and from this lambda 1 we will draw another perpendicular to the graph of chlorophyll B, and from this intersection point we will be drawing another intersection to the y-axis, and this is A2. And so this A1 is the photon absorption rate, at lambda dashed for chlorophyll A, and A2 is the photon absorption rate at lambda dashed for chlorophyll B. So we have understood that for a particular wavelength there shall be different types of photon absorption for different types of pigment. In the NCERT the carotenoid graph is also given. I am just copying it from NCERT. Okay, the carotenoid graph, you have to remember the shape of the curves. The carotenoid curve looks something like this. This is the carotenoid curve. This red line is the curve of carotenoid, which is given in NCERT. It is also absorption spectrum. Okay, absorption spectrum. So basically what we have seen that, so basically TW Engelmann have observed the photon absorption rate versus wavelength, which is also known as absorption spectrum. Why spectrum? Because there is a spectra of wavelength available for different absorptions, so it is known as spectrum. Okay, absorption spectrum. And TW Engelmann has also shown the action spectrum, and here is an interesting thing here. It is here I have shown the absorption spectrum, and the previous slides, and that my previous slide, in the previous slide you can show the action spectrum here. Okay, and you will be surprised to know that the action spectrum of photosynthesis closely correlates with the absorption spectrum of chlorophyll A and B. Just look at the curve. I am drawing the action spectrum here, very small. This is the action spectrum here. This is the action spectrum here, which I have drawn in the previous slide. Is the action spectrum. Just look at the action spectrum, this graph, action spectrum, and the graph of chlorophyll A and chlorophyll B. I am overlapping this graph. Okay, I am overlapping the action spectrum and the absorption spectrum of chlorophyll A and chlorophyll B. This is action spectrum and this is the absorption spectrum. This is for chlorophyll A. This thing I have drawn is for chlorophyll A, and this I will be drawing for chlorophyll B. So we have seen the action spectrum of photosynthesis and the absorption spectrum of chlorophyll A and B is just same. What does it mean? What does it mean? If the graphs are nearly same, then what does it mean? It does mean that the photosynthesis is maximally covered by the chlorophyll A and B. So, what T.W. Engelmann has drawn a conclusion from this from these two curves that the actions the I'm writing it here. Wait. Wait. Oh, sorry, our page is filled. We have to go into the next slide. What T.W. Engelmann has observed that absorption spectrum of chlorophyll A and chlorophyll B closely correlates with the action spectrum of photosynthesis. Okay, we have shown it in the previous graphs that the action spectrum and the absorption spectrum of chlorophyll A and B is nearly overlapping each other. So what is the conclusion out of it? So what is the conclusion out of it? The conclusion out of this statement is that chlorophyll A and chlorophyll B are the most crucial pigments for photosynthesis. This was the conclusion out of these two graphs, and this conclusion was made by T.W. Engelmann. Read the NCRT. In the NCRT, this line is being given. I am reading the NCRT here. Okay. The NCRT here says that the first action spectrum of photosynthesis was thus described, and it resembles roughly the absorption spectra of chlorophyll A and B. The line is being given in NCRT. You have to understand between the lines. Okay. So this is about the absorption and the action spectrum and the T.W. Engelmann. Okay. Our next scientist here is Cornelius von Neal. Okay. Cornelius van Neal. Okay. So what has he done? He studied green and purple He has studied green and purple bacteria. Okay. He and he also shown that light that photosynthesis is dependent on light. Okay. Photosynthesis is dependent on light in which in which here comes the important statement, in which hydrogen hydrogen this is important statement I am writing, hydrogen from a suitable oxidizable compound reduces reduces CO2 to carbohydrate. Okay, and this suitable oxidizable compound mostly is H2O. We will be seeing this later. Okay, so I will be explaining the second point now. So what is our basic equation of photosynthesis? A basic equation of photosynthesis is 2H2O plus CO2 is equals to O2 plus CH2O plus H2O. Okay, this is a basic equation. So Cornelius Van Niel has studied that this carbohydrate and this O2 and this water molecule, these three have these three have O atom, oxygen atom. Among this O2, this oxygen atom, this oxygen atom, the O this O this oxygen molecule is produced from this H2O. Okay, this oxygen molecule is produced from this H2O. That means the oxygen of this H2O is present in the O2 which is evolved in this reaction. Okay, so how did he prove it? Okay, so basically he has done a radio level experiment here. What was the radio level experiment? He has taken H2O18 plus CO2 in the reaction. Okay, and he has seen that this O2 which is being evolved is also a type of O18. Okay, this is a type of O18 and this is plus CH2O, this is carbohydrate and this is plus water. So basically this is how he has proved that the O2 which has come as a byproduct of photosynthesis comes from water and not from CO2. And please carefully look at the oxidation number in this reaction. Okay, so what is the oxidation number of carbon here? The oxidation number of carbon here is plus 4. Okay, and the oxidation number of carbon in the carbohydrate is 0. because two hydrogen means +2 and one oxygen means -2, so it balances out. So the oxidation state of this carbon here is 0, and here the oxidation state is +4. So the carbon is going from +4 to 0. So this is basically reduction. Okay, and see here carefully, the oxidation state of this O atom here is -2, and the oxidation state of O atom in O2 molecule is 0. Okay, so basically in this part of reaction it is oxidation what is happening. Okay, so let us come to our previous statement, what we have written in our previous slide. So previous slide I have written that photosynthesis is dependent on light, of course, in which hydrogen from a suitable oxidizable compound reduces CO2 to carbohydrate. Okay, so note this statement down, or I will be writing this statement in the next slide. So I have written in the previous slide that hydrogen from a suitable oxidizable compound reduces CO2 to carbohydrates. Okay, so see what is the reaction. Reduces CO2 to carbohydrate. Look, CO2, the carbon is happening going through reduction here. So this statement is verified that reduces CO2 to carbohydrate. CO2 is reduced to carbohydrate. Okay, so what is the suitable oxidizable compound here? I am talking about this. Okay, what is the suitable oxidizable compound? So oxidation is happening in case of H2O. Okay, I have shown you. So the H2O here is suitable oxidizable compound. Okay, so I was told you previously that the H2O is the suitable oxidizable compound in case of photosynthesis, because oxidation is being happening, because the oxygen is going to -2 to 0. This is oxidation, so this is a suitable oxidizable compound because oxidation is happening in case of oxygen atom. So have you understood the statement? Understood the statement is given in NCERT that H from a suitable oxidizable compound reduces CO2 to carbohydrate. It is the deep explanation of the statement. So basically the equation given in NCERT is this: 2H2A plus CO2 is equals to 2A plus CH2O plus H2O. So basically these two equations are same, because I have radio labeled this O18 and they have given this O18 is as A, because this A can also be sulfur. Okay, then when the sulfur is H2S is used as substrate, then 2S will be produced, then sulfur will be produced, oxygen will not be produced. More or less the more or less this process is same. In where the oxygen is evolved there is H2O, and where the sulfur is evolved there is hydrogen sulfide, or there might be many suitable oxidizable compound which is undergoing oxidation, and this oxidizable compound and very interesting thing, this oxidizable compound is acting as here as reducing substance. Okay, reducing substance, because basically it is reducing the carbon from +4 to 0. Okay, this suitable oxidizable compound is acting as a reducing substance which is reducing the carbon dioxide to carbohydrates. Okay, so in our next slide we'll be covering about the various pigments which is given in NCERT. Okay, so this is the next slide. We'll be covering pigments. Okay, pigments only, which is the data about pigments which is relevant to our NEET examination. Okay, the pigments is separated. The pigments are separated by paper chromatography technique. The technique in which the pigments are separated is known as paper chromatography. This data is given in NCERT. And the another data which is given in NCERT is that chlorophyll A is a pigment which is bright or blue green in color. These colors are given in NCERT. I am just citing it out. You have to remember the colors. The color could be asked in any moment in NEET examination. You have to remember it. The chlorophyll B is yellow green in color. Okay, and the xanthophyll is and the xanthophyll here is given as yellow. Xanthophyll is given here as yellow, and the carotenoids and the carotenoids and the carotenoid the color of the carotenoid is yellow to yellow orange. Okay, so you have basically in NCERT these four colors are given. These four colors are given about these four pigments. You have to remember the colors here. Okay, so in the next slide we will be covering the structure of a chlorophyll in short. We have read the structure of chlorophyll is given in the cell chapter. You can also read it from there, because in there it is given, it is in brief description. Okay, so wait a second, let me change the slide here. Okay, so here our new topic is structure of chlorophyll. Structure of chlorophyll. Okay, structure of chlorophyll. Wait a second. Structure of chlorophyll. No, no, leave it. Structure of chlorophyll. sorry, this is not structure of chlorophyll, this is actually structure of chloroplast, which is present in cell, okay, in which chlorophyll is present. The structure of chloroplast. So this is outer membrane here, and this is the inner membrane of the chloroplast. Okay, this is the inner membrane of the chloroplast, and this is the thylakoid here, which is the thylakoid. The sac-like structure present in the chloroplast is known as also thylakoids. This is the individual structure is known as thylakoid, and this whole unit of a thylakoid, whole bundle of a thylakoid is known as grana. We have to remember that grana is a plural form, and in singular it is known as granum. Okay, granum. So basically this is thylakoid and this whole unit is grana, and there are another grana where thylakoids are present, and these grana are linked by a connector. This grana is linked by a connector. This connector is also known as stroma lamellae. This connector is also known as stroma lamellae. Okay, so basically what we have learned that in the chloroplast there is a outer membrane which is more permeable, and the inner membrane which is less permeable. The outer membrane is more permeable and the inner membrane is less permeable here. Okay, and in the chloroplast there is a sac-like structure which is known as grana, and together grana constitute the sac-like structure is called as thylakoid, and a stack of thylakoid is known as grana, and the two granas are together linked by a common connector which is known as stroma lamellae. Okay, and you may also know that in the in the chloroplast there are starch granules which is stored as food. This is starch granules. Okay, and there are some lipid molecules also. There are some lipids molecules also. This is secondary, this is not important. Okay, this is the basic structure of the chloroplasts. Okay, so in the next slide we will be discussing about the light reaction, or what is known as photochemical phase. Okay, so in this slide in this slide In this slide we'll be looking at light reaction, or it is also known as photochemical phase in photosynthesis. Okay, so what happens in light reaction? I'll be explaining very short. Light reaction, light absorption occurs. Secondly, H2O H2O splitting occurs. Thirdly, high energy high energy chemicals like ATP and NADH NADPH is produced. And fourthly, O2 is released. Okay, so this is the basic steps of the light reactions. Okay, so there are photosystem which occurs in the light reaction. So basically when sunlight falls in the green leaves, the light absorption occurs, and in the presence of sunlight the H2 splitting occurs, and the high energy chemicals like ATP and NADPH is produced, and the O2 is released. This is known as light reaction. And you may know that light reaction is completely dependent on light. In the absence of light, light reaction cannot be occur. Okay, this light reaction is completely dependent on light. Okay, this is an important point here. This is an important point here. Is completely dependent on light. So in the next topic, in the next section, we have some different kinds of photosystem. Photosystem. So basically, what is a photosystem? So a photosystem. So a photosystem is is a is a type of machinery which happens which happens in different in different types of wavelengths. So basically what I'm trying to say that photosystem are different photosynthetic units functioning each at different wavelength at its maxima. So basically what I want to say is there are different types of photosystem which which operates in different types of wavelength. Suppose we have photosystem or PS1. The PS1 functions at optimum at 700 nanometer wavelength, and PS2 it functions at its optimum at 680 nanometer wavelength. Okay, so basically, have you understood what I am trying to say? That the photosystem are different photosynthetic unit which operates at its maxima in different kinds of wavelengths. So basically, photosynthesis 1 is a photosynthetic unit which operates in 700 nanometer, and the photosystem 2, there is another, this is another photosynthetic unit which 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 operates in its maxima in the wavelength of 680 nm. So these are two different types of photosystems. Okay, okay. So in the next slide we have an important thing, that is okay, in the next slide we have different types of different and interesting thing, that is these photosystems are named as their sequence of discovery. These photosystems are named as their sequence of discovery. That means if a photosystem which that means that this photosystem 1 which operates at 700 nanometer is first discovered, and then discovered is the photosystem 2 which operates at 680 nanometer. It is discovered in the second place. Okay, so this point is given in NCERT. And this photosystem 1 has a reaction center which is chlorophyll A. What is a reaction center? A reaction center is that part of a photosystem in which the maximum amount of photosynthetic activity is being occurred. Or this is and the reaction center is basically that thing also where the whole energy from the other pigments are transferred. Okay, and there are also some light harvesting complexes, or it is known as accessory pigments like chlorophyll B, carotenoids, etc. Okay, these light harvesting complexes passes the energy in the reaction center to complete the photosynthesis. To to complete the photosynthesis, I will be explaining this topic in very short. And this is photosystem 2 also has a reaction center which is chlorophyll A, and it also has a light harvesting complex or accessory pigments which happens to be chlorophyll B, carotenoids and other pigments. Okay, so this basic light harvesting complex is also known as accessory pigments also, accessory pigments also. And the function of the accessory pigment, or the also known as light harvesting complex, is that they transfer the energy to chlorophyll A, which is reaction center. Thus, they make sure that maximum portion of the available wavelength is utilized in photosynthesis. So what I am trying to say is, suppose chlorophyll A chlorophyll A shows its maximum amount of photosynthesis at lambda 1, and this is carotenoid. It it shows its maximum amount of photosynthesis at lambda 2, and suppose this lambda 2 wavelength is also available with the lambda 1. So chlorophyll A will be optimally photosynthesizing, and the carotenoid will also be photosynthesizing. And suppose if only lambda 2 is available, then what will happen? Then this carotenoid will be functioning, this chlorophyll A will be not optimally functioning, it will be functioning but not optimally functioning. So the carotenoid will transfer the energy from to chlorophyll A there. So basically what it does, it the light harvesting complex or the accessory pigments A allows plant to photosynthesize in a very large spectra of wavelength. Have you understood my point? What I'm trying to say? I'm trying to say is the carotenoid or the chlorophyll B is photosynthesizing on a different wavelength than chlorophyll A. So in addition to the chlorophyll A wavelength, they are also contributing to the photosynthetic ability of the plant. Thus, they are maximizing the photosynthetic ability in different wavelengths. That means if the suitable wavelength for chlorophyll A if not present, the plant will also photosynthesizing in a very appreciable amount, because the accessory pigments will be transferring the energy to the chlorophyll A. You have understood my point, right? Okay. And the second function of the accessory pigments is accessory pigments prevent photooxidation of chlorophyll A. So basically, if the lambda is very small, the chlorophyll A get photooxidized, and if a chlorophyll A gets photooxidized, the photosystem will be non-functional. Okay, so this photooxidation is prevented by accessory pigments. Okay, this is an important point. This photooxidation is prevented by accessory pigments. Okay, this photooxidation is prevented by accessory pigments. This is the main point here. Okay, I hope you understood my point, what I am trying to say. So what is basically happening? I will be summarizing the whole scenario in very short. This is chlorophyll A, and these are around is accessory pigment. This is suppose chlorophyll B. This is suppose chlorophyll B. Sorry, and this is suppose carotenoid. When sunlight falls into this, they will be transferring energy to the chlorophyll A. Okay, they will be transferring this energy to the chlorophyll A, and this chlorophyll A will be a part of photosynthesizing. Chlorophyll A will also photosynthesize, will also absorb energy, and the absorbed energy by chlorophyll B and carotenoid will be passed to the chlorophyll A, and then the energy will be flown in a different direction. We'll be talking about in very short when the process of photosynthesis will discuss in very short. Okay, so the accessory pigments, this is light harvesting complex or accessory pigments. They transfer the energy to chlorophyll A, and from chlorophyll A the energy is transferred to the primary acceptor. The primary, sorry, this is not primary acceptor. This is not primary acceptor. This is this energy which is gathered together in light harvesting complex and chlorophyll A will be used somewhere. We will be discussing very short. Don't worry. Okay. So you have understood this. Okay. So we will be taking some important points about the photosystems here. You have left the important points about photosystems. Okay. Important points about photosystems. Okay. The important points about our photosystems is when we talk about photosystem 1 and photosystem 2. I have told you that photosystem 1 is functioning at a wavelength of 700 nanometer, and the photosystem 2 is functioning in the wavelength of 680 nanometer. Okay, and the PS1 is situated outside the thylakoid membrane. What is thylakoid? Thylakoid is the sac-like structure present in the chloroplast, and the thylakoid, when sacked together in a unit, they are known as grana, and different grana are together linked by stroma lamellae. Okay, stroma lamellae. Okay, and the fluid in the matrix which is present in the chloroplast is known as stroma. Okay, the fluid in the uh, chloroplast is known as matrix. Okay, so PS1 is situated outside this thylakoid membrane, and PS2 is situated on the inner surface of thylakoid membrane. It is the first important thing here. You have to remember this point. These points are very important. Okay, and the second point is here is the photosystem 1. The photosystem 1 is functional at both grana and stroma lamellae, and this photosystem 2 is functional or found, same thing, functional or found in stroma lamellae only. This lamellae might not be visible. I am writing here stroma lamellae only. So this PS1, the photosystem 1 is functional. This PS1 is found both in grana and stroma lamellae. Okay, so it is found in both in the sac-like structures and in the linkers, and the photosystem 2 is found only in the linkers. Okay, and the photosystem 1 is participate in the splitting of water, and the photosystem 2, sorry, this photosystem 1 is not participates in the splitting of water. I will be explaining the whole process, because in the cyclic photosynthesis, in the cyclic photosynthesis, this photosystem 1 is functional where H2O is not requiring. I will be explaining this in very short or in the next video. This photosystem 1 is not participating in the splitting of water, and the photosystem 2 participates in splitting of water, and it is also associated with release of O2, because normally when the water is splitted, O2 is born as a byproduct. So PS2 is participates in the splitting of water, and it is also associated with the release of O2, and PS1 is not participating in the splitting of water. It is the main points here. Okay, I will be explaining. And also remember another important point, that photosystem 1 participates in both cyclic plus non-cyclic electron transfer, and this photosystem 2 is involved with non-cyclic flow of electrons. So basically you have to remember this whole chart. I will be explaining everything what I have told in this chart, that what is the cyclic process, what is the non-cyclic process of electron transfer, and what is the splitting of water and release of O2 is happening, and in what type of photosynthesis and how it is working in the next video. But you have to ultimately remember the formula, the remember these things, because these things are very important and these things are frequently asked in the exam. Ultimately you have to remember it, but I will be explaining everything what I have told in this slide in the next lecture. So don't forget to remember this chart. And also I will be explaining this chart in the next lecture, that what is the cyclic and what is non-cyclic, blah blah blah. Okay, so thank you. This is for today. I'll be explaining a very important topic in our next video session. Okay, thank you, and please subscribe to my channel for new amazing content which are related to NCERT. I only focus on NCERT. In what basis the NTA is setting the questions nowadays. Okay, so stay tuned for more amazing NCERT based studies which is relevant to the examination's point of view. Thank you, and subscribe my channel and press the bell icon here. Bell icon here. Sorry, here. Thank you.